It's just great for us to have Dave Arnold here visiting Mizzou today in the PM&R department and other parts of the university. I've known Dave since he started his neuromuscular training years ago at Ohio State. I'm an Ohio State product too, and so we've had the same mentors. He went on to do a lot more interesting scientific stuff than I ever did with the mentors. So he's really just been a rising star in the field of muscle disease internationally. And when I asked him last summer after I got here, would he ever consider making a trip out here and looking around, he said he would, but we were in the middle of COVID. So, so now he's here. So thank you, Dave, for coming. Thank you, Joe, for hosting.
Of course, yeah, my pleasure. All right, thank you, Dr. Barrett. Well, good morning, everyone. It's my great pleasure to introduce Dr. Dave Arnold today. I've got a few things that I've written down about his professional accomplishments here, so I'm going to give him a formal introduction real quick. We're going to get started on a lecture here this morning, and he's going to talk with us. And then after that lecture is over, we're going to spend a little bit of time, and he's going to share with us a little bit about his life journey. So that'll be an exciting time and maybe an opportunity for conversation and with our residents, a chance to hear, how does it play out for somebody in a career and a life? So I think that'll be a really good extra opportunity to have him share that with our folks here today. So it's great to announce you as of May as a clinical professor in the Department of Neurology at Ohio State University. Dr. Arnold did his medical education and residency training at the University of Louisville in 2004, graduated medical school 2008 for residency, and then he went on to do a neuromuscular medicine fellowship at the Ohio State University. Thank you, Dr. Conway. He has board certifications with the ABPM&R and a fellowship certification in neuromuscular medicine. And latest information on that is there are 95 people with neuromuscular fellowship certifications in the U.S. So he is in a rare crowd there. Also a fellow with the American Board of Electrodiagnostic Medicine, and he has memberships in the AAPM&R. He's a diplomat in the Association of Academic Physiatrists and participates in the RMSTP. It's a lot of letter alphabet there. It's a research medical scientist training program. Got through that one, and a fellow in the AANEM as well as in the American Academy of Neurology. And he has clinical appointments in the Department of Neurology at Ohio State, the Ohio State University, and also in the Department of PM&R there. I always have to put in the D. He's the co-medical director of the Ohio Musculoskeletal and Neurologic Institute at Ohio University, has clinical interests in functional and rehabilitation aspects of hereditary and acquired neuromuscular disorders and age-related loss of physical function, myotonic dystrophy, spinal muscular atrophy. His research interests include mechanisms and treatments for age-related motor neuron and neuromuscular junction degeneration and failure in aging and sarcopenia, peripheral nerve regeneration, effects of survival motor neuron function, biomarkers of neuromuscular function, genetics and associated gene therapies in neuromuscular disease, research grants as co-PI and PI with the NIH, NINS, biotech companies, and pharmaceutical companies, multiple local and regional and national lectures that he's done in these areas in neuromuscular disease. He's cited in multiple peer-reviewed journals and book chapters in neuromuscular disease, served in national organizations in various roles. Please welcome Dr. David Arnold.
So it's really a great pleasure to be here. Not sure where to look with it. So I gave a talk a few months ago in age-related loss of physical function, and honestly, that is my primary interest. But I didn't want to give you that same topic again. And so today I'm going to talk about spinal muscular atrophy. I work in a lot of different areas. This is basically the area that I kind of grew up as a scientist, and so I won't go into my career trajectory too much because I thought it might be fun to give you a little bit of a highlight of how I did that after this talk. So I'll try to stick close to the hour for this talk, and then we can cover that later. So I'm going to talk about kind of where we are as far as the field. It's been a lot of progress over the last over close to 30 years, and so from gene discovery to the first therapies in the clinic was just a little over 25 years, which I think is pretty amazing. And now we currently have 3 therapies that are very, very successful, but I would say that there's still some pretty significant ongoing needs that I'd like to highlight, just like finger on the pulse of the field, kind of what I think are like key areas that are really still pretty major needs for patients. So I do have a few disclosures. So some of the work that I'll talk about today has been funded by NIH, as well as a small biotech company, NMB Pharma. We just published a couple papers that looked at the effects of nusinersen in adults with SMA. That was funded by Biogen, and then a several consultant board for a lot of different companies. So broadly, the objectives. So part 1 is actually going to be a little bit shorter. So this is going to bring us up to speed on where we are in the field. And then part 2 is where I kind of want to go through some specific areas that I think are still ongoing questions that need to be answered. There's actually quite a few controversies, which is always kind of exciting in science. And then the patients still need several major things that we need to work on. So some of this might be too fast if you're not familiar with SMA. So if I go over some, don't pay too much attention to all the graphs that I show. I'll try to just give you a high-level view, but if there's something you have questions about, please stop me. So spinal muscular atrophy, when we say SMA, we a lot of times are talking about one particular form of alpha motor neuron loss in the spinal cord related to a genetic mutation. But when we say SMA, a lot of times we're talking about just these SMN or survival motor neuron related spinal muscular atrophy, but it's actually a heterogeneous group of disorders. There are distal SMAs, there are proximal SMAs, and then there's also scapuloperoneal SMAs. But we're only going to focus on one genetic form today, and that's the one that we really have the most progress in. So clinically, this results in alpha motor neuron dysfunction. It is in my opinion, a remarkably restricted phenotype just to the motor system. So you have effects of muscle weakness, muscle atrophy, reflexes are reduced. A lot of times patients with more mild forms of the disease have a very striking tremor, which people call polymania myoclonus. My thinking is that's related to denervation, renervation. So the motor unit control, you've lost that perfect coupling, and so the physiologic tremor is amplified. And this is a pretty classic phenotype, but what historically has made it challenging is there's actually a range of severities. And so this is related to one gene loss. So it's a homozygous loss of the SMN1 gene or survival motor neuron 1 gene. That is usually a large deletion, but it can be a compound heterozygous state where you have deletion with a small missense mutation. And if you look at all forms of SMA, this is by and large the explanation for the heterogeneous group. This explains most SMAs, but there are also non-SMN SMAs. There's a second gene that is present in all patients called survival motor neuron 2 gene, or SMN2. And this is universally present in all patients, but it is variable in the copy number. So patients may have 1 copy, but they may have 5 copies, or a range of copies up to even more than that. And as you have more copies of SMN2, you have a less severe clinical phenotype. So later onset, less severe muscle weakness, and less severe comorbid complications related to the disease. And so why is that? And so these 2 genes are very, very similar. Actually, there's just a single nucleotide change between the 2 genes that make SMN2 act differently than SMN1. So basically, SMN produces essentially 100% of full-length transcripts of this gene. So then you get a full-length protein which acts and functions normally. SMN2, because of this C to T transition, you actually, in the majority of the transcripts, you don't have this exon 7 included in the transcript. So it's a shortened, truncated version of the transcript. So that makes a shortened, truncated version of the protein. So 85–90% of the transcripts from SMN2 in the untreated state produce this protein that has some function, but not much. And so basically it just rapidly disappears and it doesn't really work that well. So SMN1 is gone in patients with SMA. And then SMN2 is present in variable copy numbers. And so as you increase the number of copies, then you get more of small amounts, but more of this normal functional protein from the backup gene. Interestingly, humans are the only vertebrate species that have 2 SMN genes. Chimpanzees actually have duplicated SMN1, but no other species have an SMN2. And universally in all systems that this has been tested in that have a SMN gene, when you delete that, it's not compatible with life. So you need some SMN to function for survival. But what's, I think, one of the most interesting things about SMA is when you have low levels, it doesn't cause problems in all cells. Well, that's one of the controversies that we are going to talk about. What did I do here? For whatever reason, you have this remarkably restricted motor phenotype when you have low levels from the SMN2 gene and absent levels from the SMN1 gene. So it is a motor neuron disease, despite the fact that this protein is expressed everywhere. The expression level does change during development dramatically, and we've studied this. We've shown that it actually goes down during aging, and so that's some of the work I'll talk about today is actually from the perspective of aging and age-related loss of physical function and the effects of higher levels of SMN. So currently, there are 3 therapies that are available in the clinic. I was mostly involved in the gene therapy approach, and so some of the data that I'm going to show is going to be more specific to that aspect. But the first therapy that was approved was the nusinersen. It's an antisense oligonucleotide that basically makes the SMN2 gene act more like SMN1. So it turns this wimpy backup version of the SMN gene into a better version like SMN1. So you get more full-length transcripts, so more normal protein from this backup gene. And really, the small molecule compound, which actually was the 3rd to be approved as of just a few months ago, it's an oral compound, an oral drug that people take. It does the same thing. It's just using a small molecule approach. It actually binds a couple different players in splicing to have a very specific effect. And so the nusinersen is an oligonucleotide, so it's to a sequence that basically encourages the inclusion of exon 7. Risdiplam is a small molecule, does the same thing. And then the other strategy has nothing to do with SMN2. It is to basically replace the gene that's missing. Replace is a little bit of a strong word, so that's the reason I put it in quotes. So it's actually delivering the cDNA of the SMN gene, and it's not integrated in the genome, which is a good thing because you can disrupt other oncogenes and things like that. But in essence, it's putting the cDNA into cells, and then that produces the SMN. What's kind of interesting, a lot of people don't know, the cDNA that is actually in the gene therapy Zolgensma is actually SMN2, because the reason that is because it was available. My mentor actually had this, and so it's what happens. What causes SMN2 to not act like SMN1 has to do with splicing. So if you put the cDNA in there, then that's a non-issue. And so that's the reason it acts just exactly the same. It's just the difference is splicing of the SMN versus SMN2, which I think is kind of a cool little story. Most people probably don't know that. I sometimes think they don't want people to know because then it causes questions, but it's exactly the same. The genes are exactly the same. So when you put it in a cDNA. So those are the 3 therapies. Not all of these work for all patients. So gene therapy limitations are size because it's purely a generation of virus. And so the strategy to make it more applicable to older individuals is to do it in the spinal fluid, and that's kind of in the progress. There's been some hangups with that. But currently, this is a systemic delivery, so it's a lot of virus, and so there are age and size limits depending on where you are as far as who can get this. The first 2, the nusinersen was the first therapy approved in late 2016, and it's actually approved for all SMA patients. The challenge is it's an intrathecal delivery, and you have to do that repeatedly, and patients with SMA oftentimes have complicated spines, so access to the intrathecal space is really challenging. But this is approved across the board. And then the risdiplam was approved after 2 months of age because they simply didn't have data in younger age yet. And so I eventually think that will change. I actually don't know the status of where that is. But so ASO is kind of universal. Risdiplam is universal after the first short period. and then gene therapy is only very early. And so there's a lot of caveats when you think about which one you want to use here, as far as, delivery and things like that. And one thing that we'll circle back in a little bit is, remember, SMN's low everywhere. So if you deliver it in the intrathecal space, you're not increasing it everywhere. We don't know if that matters. Personally, I kind of don't think so, but we currently have a grant right now to test that. And then gene therapy is not integrated. So if you deliver it and it transduces a dividing cell, you're going to lose that as the cell divides. And so if high levels of SMN are needed in a dividing cell population, that may not have the effect you want. The good thing is motor neurons are post-mitotic, so they don't divide. You can't replace those. AAV9 has particularly good tropism for that cell. And so it does go to that cell and it should persist, but very, very long-term studies, we still don't really have those in humans. So will this become inactivated? Those are still some big questions. Other supportive care. So this is basically managing the secondary complications of musculoskeletal. So patients get contractures. They also have very dramatic hip instabilities and have hip dislocation. Patients' fracture risk is extremely high. We had one patient that simply just being transferred to have fluoroscopy because of complicated spine to get the noose inertia had a femur fracture. So it's very, very challenging positioning and mobility. And then spinal deformity, I think, is becoming more of a challenge in some of the earlier treated patients because there's a lot of axial weakness, like SMA type 1. Historically, the mortality on average is around 10 months. And so they never really got the spinal deformity because they weren't sitting. But now that they're kind of transitioning to more type 2 or 3, they're sitting more, so there's more axial problems. So these are areas where I think rehabilitation, like these are the patients, as we have more and more gene therapies that come into the clinic, it fixes a lot of problems, but it actually makes new problems happen. And so being able to see the whole patient together, understand where the function is, where the problem is, and what tools we have to help them, I think is a really exciting area. And I think this is just one disease, and this is very rare, but this is I'm really involved in myotonic dystrophy. I think that's the next step, one of the next ones to have a good success, I think. So I think this is a big area of opportunity as these get translated, understanding new problems to use our skill set for these patients. So now to the meat of the talk. So I have 3 main domains I want to talk about where I think there is gap. Now some of these are more answered than others. This first question or concept is timing of treatment, and this was actually probably what got me into spinal muscular atrophy because I was very interested in this. My first career development award through the RMSDP, through the AAP, was to answer this question using mouse models. It ended up during that same time frame. I was involved in mouse work and pig work and also the first human study. So we actually kind of saw it in multiple different scenarios. But this is the first question I want to address. What is the right time and how early is early enough? The next one will be delivery of treatment. So how much and where? And then the last is, I just submitted the paper on this yesterday, resubmitted it, on what can we do to develop new therapies. So not related to SMN protein restoration, but like what I would call additive or like combination treatments. So this is an old graph, but was kind of like a big part of what kind of was my question as a early scientist is what is the natural history of motor neuron dysfunction and loss in SMA? So this is a disease that it's a little hard to tell exactly when it starts because infants are developing so fast. So you're looking at a moving target, and then you also have this disease on the background. So using more objective measures of what I would call motor unit connectivity. So the motor unit is a motor neuron and all the muscles it connects, and each muscle has, a couple hundred motor units or so, depending on the muscle. And you can measure these with electrophysiological measures that we do in the clinic all the time. So CMAP and modifications of CMAP, motor unit number estimation. So this is like this is a combination of a couple different studies. So Dr. Finkel put this was really just about one patient, but it was on the backdrop of 3 previous patients from a study by Kathy Swoboda. And it showed an interesting thing. And so it's looking at age, and then you're looking at either electrophysiological measures or a clinical measure of motor function. And you see that they have this, even though they're very young and early in their development, they have more function, and then they have this kind of fulminant drop. And I've been involved in multiple biomarker studies and then intervention studies, and I remember one case in particular. Her sibling was diagnosed with SMA, so I actually ended up doing CMAP at like 11 days of age. And I remember her CMAP was like 6 millivolts. So here, this is in microvolts, just to clarify. Her CMAP was like 6 millivolts. On average, when you look at like previous studies, on average, chronic SMA CMAP is around 0.2. So that you're talking about a huge magnitude of drop. And what was sad is she was actually one of the early participants in the gene therapy study. So she actually pulled herself, or her parents pulled her out of the biomarker study to enroll in the gene therapy study. And her baseline measure for the gene therapy study, her CMAP was 0.3. So you see this, like there is like some point during early, shortly after birth, where CMAP seems to be preserved. But there's still a lot of debate of whether it's developmental or degenerative. But my concept for this was like, is there a pre-symptomatic treatment window that we could target, which is actually still quite debatable. And so one of the first studies I did, and this was part of my RMSDP, and I won't go through this too much, but basically the Delta 7 mouse model, my primary mentor for my career development grant, and we'll talk about that after this, he created one of the first mouse models. This one actually was a subsequent model that we used, but it lives about 2 weeks after birth. It has the human SMN2 transgene, because mice don't have SMN2, and then both of the mouse SMN genes are knocked out, basically. And then it also has something in there called Delta 7, which is basically the cDNA of the SMN2 with exon 7 not included. And these mice were felt to be one of the, and still probably are one of the best mouse models of SMA, but there's a few other mouse models currently. But what I did first was map out with electrophysiology how the motor unit declined. Did we have a pre-symptomatic window? And I found that things seemed to change about 6 days, so not pretty early. So that's really taking these measures down to like a small, tiny mouse. And then that was the first study, and we treated early, and we showed that really if you treat early, mice seem to look pretty normal if you treat like on day of birth. And then subsequent study was to delay the treatment and see the effects. And basically, when you boil it down to gravy, you basically have diminishing weight gain. And so you can see, like, as you delay the treatment, we have untreated, treated at 4 days. That's kind of before the CMAP dropped in these animals, but still kind of delayed. And then 6 days. And you see this just diminishing decline on weight and survival, as well as electrophysiological measures of motor unit number and CMAP. So very simple measures, but things that we could do in patients. And that was kind of my angle, is I wanted to see, like, I was actually surprised nobody had done this in mice. It's like something we do all the time in the clinic. Why not? And so this actually was a pretty exciting result, because it suggested that we can get good effect if we go in early. But it also, I was worried with this data when we were planning the gene therapy study, which was right around the same time. It's like, well, are we going to have any effect? Because we were going to be enrolling SMA infants at very late stages, as far as way after their CMAP had dropped. So I kind of predicted that we might not see a big effect. And so one thing I would point out, I've studied SMA models across mice. We have a pig model and also SMA patients. And as I mentioned, you can see this is the healthy control, and this is the SMA1. So this is 2 copies of SMN2. These are 3 copies plus. So this could be, a less severe form of SMA. You can see that there's a kind of a dose effect there on the CMAP. But one thing I want to point out here is actually the mice aren't that severe. So this is a mouse that dies in 2 weeks, but their electrophysiological measures are really less than 50% reduced, where when you look at humans, it's more like 95% reduced. The pig's kind of in the middle. And so I'm going to just show you just a little bit of the pig data. So this is a unique model because they don't have SMN2. I know Dr. Orson was in the crowd. has been working on an SMN2 transgenic pig, which is really exciting. This strategy, what we did is we reduced the SMN levels in motor neurons by about 80% by using a knockdown approach. So one viral vector knocked down the pig SMN, and then the other viral vector was replacing the SMN protein, human SMN protein in the pig, and to see the effects. And what we did is we basically mapped this out with early treatment, so we basically knocked it down and replaced it right after, or we waited until the pigs developed clinical weakness. And their phenotype showed, ventral motor axon loss, fibers of fibrillations, which we would expect in acute denervation, and then you can see just the decline of CMAP and motor units in the untreated pig there. And this is just a quick video of a control that they run up this ramp to get either sweetened condensed milk or marshmallows. And you can see that the SMA pig is quite weak, more in the hind limbs than the forelimbs, which is similar to patients with SMA. And we do eventually still give them the condensed milk and the marshmallows. They don't get that kind of stuff. And then early rescue and late rescue. So this is knockdown one day, rescue the next day. And you can see looks pretty normal. And then late rescue, you can see not as normal, but still normal. So you can still see some slipping with the hind limbs and splaying. It's pretty subtle. Sometimes it's hard to see the motor phenotype of these animals, but very interesting that even with the late treatment that we got an effect. I would say even maybe more robust than what we saw in the mice. So I thought that was encouraging. And when you look at electrophysiological biomarkers, which we do in the clinic, you can see it basically looks like what you would expect. So CMAP is a complete readout of the functional status of a muscle. So you can see it's reduced without treatment, and then it's normalized with treatment. So because the motor units can collaterally integrate the muscle fibers, they can kind of compensate for the output. Because when you look at the number of motor units late treatment, you do have losses of functionally connected motor neurons to the muscle. But it seems like it kind of normalizes with this collateral sprouting. Same thing, this is anatomically using morphological counts of either the alpha motor neurons in the spinal cord or the number of ventral motor axon counts in the roots. And so it shows that mouse, early, late, it's very time sensitive. So I guess that's not surprising, but I think it was a really important finding for the field. And so with this, as this was coming together, I was very curious what we would see in the clinic as far as the effect on timing with patients, because the way that this works, so FDA is changing their strategy for a lot of these trials. Actually, I've been working on a myotonic dystrophy trial where the plan was to go into healthy controls. It's also an ASO, but it's antibody conjugated to knock down the gene that's has the repeats in it. And the plan was initially to go in healthy controls, but then FDA didn't want that. So in DM1 study, going into DM1 patients, same thing here. Like this is a gene therapy, you're delivering a gene. You can't test this in healthy infants. And so the plan was initially to do 3 different doses, a low dose, an intermediate dose, and a high dose. And it was all going to be symptomatic SMA patients. So I didn't really know what kind of effect we would see. And I'm not going to go through this in too much detail, but what I wanted to show is, so one of the infants that I mentioned that I saw in the biomarker study, unfortunately, was in one of these first cohorts, was treated very late with a lower dose. Didn't have a great effect, but did have some effect. But you can see this actually is the intermediate dose. We never went to the high dose because there are some side effects of the treatment where liver enzymes can go up, and generally infants are treated with steroids to prevent that. But one thing that I think is like very, very interesting is you can look at the timing of the treatment here. So this is distributed by age when they were initiated on treatment. And you can see there's a clear time-dependent change. Most of the individuals that were treated early, even if their function was crazy low, I mean, look how low this one is and how high it goes because it was treated on the earlier end of the timeline, which I think is fascinating. And most of these individuals had CMAPs that were decimated. There were a couple, these 2 here actually were treated quite early. So one was at about 4 weeks and the other was at 6 weeks. The one that was a little bit lower, actually, she dropped before she actually got her treatment. It was kind of interesting. This is just a little personal story, but her family stayed in the Ronald McDonald house while she was in this trial. And my sister's daughter had epileptic surgery at the same time. So my mom actually held this baby, and they didn't make the connection until after the fact. It was pretty cool. So now if you look up like Avexis and Novartis' website, you'll see pictures of all these kids that are like superstars. Just
Why was the decision made to even let older babies in? Why didn't they just restrict it to young babies to begin with?
well, because it was kind of the risk benefit. Because we do kind of know the natural history of SMA, but these were all 2 copy. So we very much predicted they'd be type 1, but they really wanted the patients to be weak. Yeah, so it's more FDA thing.
Okay.
Yeah. So, but clearly you can see that there is a time-dependent change, and this was all one center, all open label. So you got to take that with a caveat. There's many more patients treated now.
What do you think happened with patient 8, the one that was started at like 6 months? It's like this one, kind of improved for a bit and came back down to baseline and then back up.
Probably noise in the measure, probably just stable. So the natural history is normally like a decline. So I think it's just stabilization, probably just quite severe. Yeah.
Any data points that you don't have there that extend out the age?
There are, but I didn't put that in.
Right, any comments though? that you-
I mean, so-
Sustainability.
What's the mortality in treated patients?
Oh, it's like 95% by
Yeah,
2 years.
right,
Yeah,
so
yeah. But so some of the individuals subsequently gained the ability to stand. I think they are all eating by mouth, except maybe 1 or something like that. So they did continue to get better. And some of that's like, I think just development on top of the disease. So how much of that is SMA treatment? I don't know.
Right.
But I could not believe when we saw these kind of effects
Yeah.
because I predicted these early kids. So my hypothesis, and we wrote a review about this in 2013. My hypothesis was if you go in early, you're going to have a good effect. And it's going to stabilize. But this like resurrection effect, I don't know what that is. There is just a recent paper. I'm going to briefly talk about it, like what's happening electrophysiologically there. Because most of the studies, unfortunately, just use CMAP, and that doesn't tell you about motor unit number. And so that's one of the gaps that I think is a really big question in the field.
How long are they living with treatment?
So the longest one out now is about 6 years. So we don't know, but I mean, like this guy here, like the last time I saw him, like they did not like what I had to do. They don't like CMAP. I was good at it and I would do it quickly, so it wasn't terrible. But when they started getting older, they started remembering. So this kid, he said no, and he just ran off. He ran, and this is a kid that is,
Success.
he should. That's why I said, I was like, why are we doing these things? But I think honestly, like, we're only looking at the tip of the iceberg. So we all know about polio, post-polio, and we know that people have polio, think, I was only weak here, so I don't have it. But then you do an EMG, and it's everywhere, and then they get post-polio later. And so I suspect that they may have more weakness as they get older, just natural losses of motor units and things like that. So that's a gap. I
Do
think.
you see
Sure,
fiber
but-
splitting and things like that, like you do in polio? Do you see that in these
What's that, fiber splitting?
fiber splitting?
And what do you mean, like muscle fiber? Well, we don't do biopsies, so I don't know. We see that they have massively enlarged motor units. I'm going to show that.
That is an interesting point. You're gonna get to this at the end. One of the complications of successful therapy
New
that
problems.
we may end up seeing post-polio-like syndromes in these kids, which all would be dead.
Yeah,
And so
yeah.
this
right,
is.
true.
It's a success.
This is the biggest
We're just
neuromuscular breakthrough in my career.
Yeah.
And it's just extraordinary.
Yeah, it was amazing. So I didn't mention this, but like there were literally days where I'd do my clinical things, see consults in the hospital. I'd study mice with SMA. I'd study pigs with SMA, and then I'd truck it over to children's and see a baby with SMA. Literally all the same day. It was just like I am living the dream. This is like so cool. Walking through history, like right in front of my face. Like because with the Muni, so it never got published. That's a long story. But I was able to, we did Muni in this study. We published the CMAP, but not the Muni. I was able actually to see what was the status of these patients. I was like, wow, this is maybe the first baby that ever has been cured. And that's a stretch, but still it was like
Not much.
really amazing. Yeah. Okay. And so ongoing gaps. So we know that early is better, but how early is early enough? And there is actually a lot of question about whether this is still a developmental problem. And there's been some big papers by different groups that have shown this. So Charlotte Sumner just published a big paper where they looked at actually autopsy samples of patients, and they did a series of very nice experiments in mice to show that even early on, like intrauterine, the mice, their axons seem to be small. What that exactly means, we don't know, but they showed in a series of experiments that really to get full effect in mice, you had to treat intrauterine. So they actually treated these mice before they were born. The difference between my measures and theirs, I'm really more on the in vivo level where they're doing more selective recordings, because what we do know is different pools of motor neurons seem to be more or less susceptible. So I may have been partially measuring from a pool that's not as susceptible. So what that really means, we don't know, because like the existing data is like when a human is born, they don't have polyneural innervation at NMJ. Mice have polyneural innervation at NMJ where multiple motor neurons come into the same junction for 2 weeks after birth. So is this a developmental thing of mice? I think that's a big question. I wanted to mention this study just briefly. So this is out of Australia, where they use a different type of immunity technique that I'm not totally sold on, but they seem to show that in symptomatic children that were treated with mixed nerve syndrome, they think that there's added motor units that are occurring. I don't know if I'm totally confident in that, but this was published in a really good paper. The technique makes me wonder, and we'll talk about it in a little bit, whether the way they're measuring immunity may be artifactually impacted by the neuromuscular junction phenotype, which we have just recently described in more detail. So I think it remains to be seen like what's happening as these children get stronger. Based on my anecdotal observation of the 12 individuals that I studied with immunity, their immunity seemed to be pretty stable. It didn't seem to go up with the technique that I used. So I think it's more, we're getting more from the motor units that are still there. And the earlier you go, the more motor units you have to act upon. I don't think we're creating new motor units because I don't know what the mechanism of that would be. But one possibility could be you could have silent junctions or motor neurons that are inactivated, but I think we need a lot more research to figure that out. And so one thing that we are particularly interested in is the effect of treatment in adults, because this is the extreme case of late. And so we saw that there was effect in infants, even though they were very, very symptomatic. So what happens in an adult that's had this disease for decades? A less severe version of the disease, but has had the disease for decades. So this is mostly led by Dr. Bakri El-Sheikh, but he and I collaborate on a number of different projects, and you can see that we have come up with ways to try to deliver this where it's easier for the patient. So we have an interventional spine specialist that does a cervical injection, and we have this kind of inflatable pillow where they can stay in their chair. They don't have to transfer, so we're doing these injections upright under fluoroscopy. And so this is mainly an issue in the non-ambulatory cohort. So we had an ambulatory paper and a non-ambulatory paper that we just published within the last couple of months. And I didn't want to go through too much of the details, but basically on all the different motor function outcomes that we did, there was either stability or slight improvement. So there was improvement. Open label study, that's an observational study, not really open label, more observation, because all these people were treated clinically. What I found interesting though is we did consistently see increased CMAP, and we did immunity in this cohort. So motor unit number estimation, so estimate the number of motor units in muscle, and immunity was completely stable where CMAP and SMUP both went up. So one question that I have is, are we improving the connectivity of the motor units at the muscle, maybe collateral sprouting, strengthening of the transmission through this treatment? And that seems to be a very consistent effect. This was one of the first papers really to show that. And we showed it in both the ambulatory and the non-ambulatory cohort. So maybe we're getting more from less by restoring SM levels. So we've been doing a new technique. I don't know if anybody here does this, but it's called decomposition surface EMG. So you take this messy surface interference pattern that you've probably seen if you're doing a CMAP and somebody contracts and it's not relaxing. So this technology actually is using 4 different electrodes from different vantage points. And it's looking at the same motor unit from different angles, and using basically artificial intelligence breaks this down into all these different motor units. You get a vantage point from different perspectives of each electrode, and you can get a number of different parameters. So one parameter that you can look at is like recruitment. So this is just a healthy control that did a 20%. So we can link this to force. You can do a 20%, 50% of max contraction. You can see that you're pulling in the higher threshold motor units. Each of these lines is basically a firing instance. And so you can see these are firing like consistently here. This is not just, they didn't relax between the trials like they're supposed to. But you can see, and this is a hand muscle. So motor control is controlled by motor unit recruitment and firing rate, or rate coding, what people call. That ratio or balance between those two to control power is different between different muscles. And this is a hand muscle. And so the balance between rate coding and motor unit recruitment is going to be different in different muscles. But in the small hand muscle, you can see by the time we get to 20%, we're almost pulling in all the motor units that they activate, which I find interesting. So we've been applying this to SMA. I don't have data on recruitment yet because this data was captured actually without the load force transducer. So this was not with force. This is just like a max contraction. But what we found, which I found quite interesting, is when you compare those motor unit sizes, and this is cool because no stimulation. This is just them contracting. It takes about 5 minutes to set it up and measure it. It takes hours to get the data though. But you have them do this, and then you can get this biomarker. And what we found, which I found interesting, is the motor unit size was increased in the mild patients. So the ambulatory patients had larger motor units. And in fact, it was smaller in the non-ambulatory patients. So it may be that there's this defective motor unit number that happens with SMA, as well as the size or connectivity of the NFJ. And maybe that's what's happening with nusinersen. So we're wanting to study this in a number of different contexts. Look at excitability dysfunction, because it may be that rate coding is altered in SMA. And that, I just got a new R1 to look at that domain in aging. So we may be able to apply that to SMA as well. So could motor unit discharge be altered and drive symptoms of fatigue, or which are very prominent in SMA, and don't seem to respond to nusinersen very well. And also, like, we could use these biomarkers to track like exercise intervention, and because strategies for preventing fatigue could use motor unit recruitment or firing rates, and we may be able to modulate those with different kinds of interventions. We have shown, this was a collaboration with a group at Northwestern, that motor neurons are hypoexcitable in mouse models of SMA. This is more of a mild model that lives longer. We have a lot of data in this regard that's not published, but hopefully that'll get out soon. And this is just pointing out that fatigue is a major problem. And motor unit firing, actually, I believe, is probably going to play a role with fatigue. So not able to fire the motor unit is going to make you feel like weakness and more effort. I've always wondered if chronic fatigue syndrome has motor neuronal kind of problems in the spinal cord. I don't know if anybody's ever looked at that, but it's the kind of patient that we don't always want to see, because fatigue is like this so vague thing, it's hard to quantify, we don't have anything to do for it. But I think some of these techniques might help us understand that. So timing of treatment conclusions, times motor neuron early is better, but how early do we need to go? Aging effect, as I mentioned. And we really need better biomarkers, and we need more dedicated studies looking at very early treatment and what's the difference between very early treatment and delayed treatment, and where are the gaps. So if you treat right after birth, how sufficient? Like, do they have normal counts of motor units? I think that's a question that we still really don't know. And maybe they won't. Maybe that they won't because there is a developmental piece, or maybe our therapies aren't increasing SMN enough. I think that's also a question. So that's kind of where we go to next. So I call this kind of the Goldilocks phenomenon. So how much is enough and how much is too much? And then where do high levels need to be? SMN is everywhere, but when it's low in motor neurons, we know it causes problems, but are there other tissues that are also sensitive? And so very high profile paper that has caused a lot of distress. Yeah, that's right, and very high profile. It was just on the top. Yeah. Like right up there with New England Journal. So this was, I think, in Nature Neuroscience, just came out within the last couple of months, and actually has caused a lot of stir in the field and a lot of distress. So this, and we'll go through the data a little bit, suggests that there's a toxic effect of high levels of SMN. So now we have these hundreds of, maybe thousands of patients that have been treated with Zolgensma, and now there's a toxic effect. So it's a very stressing kind of time. So we have specifically been trying to work on some experiments to further address this question. And so basically what they showed is they actually mostly studied wild type animals, and so they treated wild type animals with an AAV to overexpress SMN. They used various dosages. Some of the doses didn't have the effect on this. They did measures of what's called rotor-rod. It's like a balance coordination measure for mice. Inverted grid is where the mouse kind of holds on. And so it's kind of like a stamina measure. And then they also looked at weight and survival, and it showed that with the higher doses, they had decline in functional readout. They also measured, so this is the M weight, so CMAP, and this is H reflex. So I don't think people have probably done an H reflex. So the H reflex gives you a sense of the spinal excitability, and it also tells you about the connectivity of the alpha-1A pair and the motor neuron. And the H reflex has actually been kind of a topic in SMA for a while, and there's some nice papers from this group looking at the sensory motor circuit. And what they basically showed is with these wild type animals that were treated to overexpress SMN, they had reduced sensory and motor connectivity in the spinal cord, so decreased H reflex. And that's either absolute H reflex or normalized to the M wave or the CMAP. So that gives you kind of an index of spinal excitability. Their work showed that they've got these very large aggregates of SMN. So when they overexpress it, it's these large aggregates. And really what they suggested is that's causing sequestration of splicing factors and machinery, and then basically, in essence, causes a functional depletion of SMN because it's caught up in all these aggregates, and that's what's causing this sensory motor deficit. They're suggesting it's more prominent in the dorsal root ganglion and the motor neuron, but you can see that these very striking aggregates in the DRG and the motor neuron. The caveats that I think they undersold in the way that they put this together, and so not everybody catches it. It's a different promoter. So the promoter drives how much this viral vector expresses. It's also, they use a higher dose. And when you compare the expression like to wild type animals, they got 2,500-fold increase. So it's massively overexpressed. And when you look even at their lowest two doses, they didn't really have these phenotypes. And so I purely think this is a dose effect, but we're trying to explore this to understand, because this is, like, a lot of clinicians see this paper and they're feeling guilty that they treated their patients with gene therapy. I literally heard that from my doctor.
This is all mice.
Correct. But even that, I mean, that's what in the mice it works, but, and it's also wild type mice. We're not going to treat wild type people. There's no such thing as a wild type person, but as wild type, in regards to basically, if you flood the cell with a lot of SMN, it causes a problem. And so some considerations: high expression. And if you look at the prior work, now the differences is one is cerebral spinal fluid delivery, the other is systemic. But in the original paper, with the vector that's in the clinic, the SMN levels of the untreated SMA were only increased about a third over untreated SMA. So treated SMA versus untreated about a third, and only about 70% of controls. So they didn't even overrun the wild type animals in that model. Now, one thing here is this is like whole tissue, and the virus hits different cells. And so, yeah.
What type of vector, as far as the nausea versus, is it just the oligosensitive? So that's the delivery mechanism itself could be causing some of the.
Yeah, so every strategy could induce different levels of SMN but the difference between the AAV and that's a really great question. AAV and nusinersen and the risdiplam is one, the nusinersen and risdiplam are acting on a target, and so they're limited by that, and it's not going to overrun that. It's not autonomous, where the gene therapy is kind of autonomous. It's going on its own. So that's the difference. So this is only relevant to Zolgensma.
So you guys think that the vector itself
Yeah,
causes the problem?
yeah,
Well, people also have looked at that, but they didn't show it in the GFP. So it's probably the vector, it's probably the promoter and the dose. So we have been doing some staining in our pig samples to see if we see these aggregates. And you do always see granular staining. So this is just immunohistochemistry. So we're staining chat is a label of motor neurons. This is human SMN because we delivered the human SMN in the pigs. And then this is merged. So normally GFP, green fluorescent protein, is what we use to show which cells were targeted. Normally it's green, but we're putting it in the same color as this paper, so it's blue here, so it's a little confusing. But basically, we don't see those big aggregates. We're seeing more granular. This is typical. And so we're now going to do a series of different doses in mice that kind of approximate what they did, but with the clinical vector to really test this to kind of put people's mind at ease. So we're trying to work with this. I also have mice that, because I have an aging grant looking at SMN, because I actually think it's protective. We've shown that it has an effect with reinnervation and preserving motor function during aging. So these are 6-month mice, the same age that they did the H reflexes in. So these mice have more physiological levels of SMN overexpression. So a couple, threefold here, depending on if you look at spinal cord or brain. And we did M-wave, H reflex, and we don't see any difference there. Also, really fine. And so this is kind of showing if you have physiological levels of high SMN, we don't seem to see those sensory motor circuit deficits. So this is recent data that we're trying to put together just to kind of counter what was published. Because I think what they did was very beautiful work. It's just the question really isn't relevant to the clinical situation. I think that's the thing. We also have looked at young and old mice. So these are color coded. I don't have 8 copy here. So 16 copy or 2 to 3-fold of wild type. And you can see maybe they even have some protection during aging. Don't know exactly what that means yet. I just wanted to toss this out here. So SMN is expressed everywhere, but it causes a relatively restrictive motor phenotype. But there's a lot of people that are wondering about other tissues. So the mice, I wish I had pictures here. When you rescue a mouse, they a lot of times have this really short, ugly tail, and as they get older, it'll become necrotic and it'll fall off, and then they become hamsters. But humans don't have a tail and don't normally develop necrosis, but there's all sorts of people trying to say that SMN is a multi-system disease. And I think we really don't know. I think in mice it is, but their motor neuron phenotype is not that strong. So one has to wonder, is there different sensitivity to low SMN in different species? And I think our models are not perfect, so we have to keep that caveat in mind. So a lot of people are making a lot of excitement from the mice. There's some studies in patients, but they're not very well controlled, because if you have severe motor dysfunction, you're going to have other systemic problems. We know in spinal cord injury, when you have spinal cord injury, you have metabolic syndrome, you have bone loss, you have all other kinds of things. Same thing happens in that SMA, and they're trying to say it's related to SMN deficiency. But to do that, you really need to compare to a control group, an affected disease control group, to see like what is the effect of motor loss versus SMN reduction. And I think there's not enough rigorous research in that area yet, but we are currently looking at effects of, so nusinersen is intrathecal delivery, and we're going to look at people as they transition to the risdiplam to see, which is the oral compound. So that's going to increase SMN everywhere. We want to see, like, does systemic delivery have an effect that's different than intrathecal delivery? And so that's one study that we got one going.
So what happens to the, like you said, it could be age protective, right? So
Yeah.
are you doing anything on mice that don't have the, like, what happens if you overexpress that? So then you give it to a normal person or a normal baby.
Yeah, so those mice are actually normal. We just added extra SMN on top of them. So they're transgenic mice with extra SMN. We're also developing a new viral vector to deliver. So AAV9 is very good at transducing motor neurons in young mice, but once they're 5 days old, it just does glial cells. And so we have a new viral vector that will transduce motor neurons in adult mice. So we're going to test it transgenically. We also want to treat old mice. So we'll age them out and treat them like this delivery, because they don't have SMN2, so we can't modulate that. So it's a little tricky. But we have several experiments going on. So, yeah.
I wanna live forever. So.
That's what all my friends said, so just take a vitamin Smin is what they say. Okay, so last concept is, just a few minutes, is the question of, are there other ways we can improve motor function? And so people have looked at myostatin inhibition to increase muscle size. So I don't know if people know myostatin, but it's a negative regulator of muscle size. It's been used in aging, other neuromuscular diseases. There actually was just a positive clinical trial from Scholar Rock using kind of one of the targeting part of this pathway just recently. So maybe this is a strategy.
Maybe mice or
Humans. Yeah, type 2 and 3. Just it was, I think it presented at Cure SMA last week.
seem to never work in.
I know. What was interesting about myostatin, we actually have studied this a lot in aging, is when you block myostatin, it makes the muscle bigger,
Yeah.
but the muscle quality goes down. So you have a big muscle that's not as strong. And that's just like more weight. So that's not really that helpful. And also, like, if you have fewer motor units, what happens to that scenario? So, yeah, so I'm curious. I don't know that data that well, but it was just published high level or presented high level. So the last part, this was an add-on kind of sub-study that we added to the 2 studies that we just published. This is the one I just resubmitted last night. And so previously, people have shown in untreated SMA patients, NMJ transmission is not working like it's supposed to. As an EMGer, we probably know when we see denervation, reinnervation, we always think the motor units are unstable. So we just say, oh, that's just denervation. Well, what's really interesting is there's more and more data to suggest actually if you quantify it, they have a lot of decrement on repetitive nerve stimulation. And so the goals of this study were to look at repetitive nerve stimulation before and after nusinersen and to see how it links up with other measures of physical function. So we did really just a simple measure. I do a lot of single fiber EMG. I actually have a couple of single fiber EMG studies going in some other diseases, but we just did simple 3 Hz repetitive nerve stimulation from the trapezius, which has historically been shown in untreated patients to show decrement quite strikingly. As you all know, 10% more loss of the size of the response or amplitude is generally considered pathological. And so this actually came out of some preclinical studies that we had been doing. So this, again, is that same mouse model I was talking about, treating at 4 days of age and showing like partial rescue. And we showed that compared to controls, they had striking NMJ defects. Now, is that because they have motor unit loss? And that's my hypothesis, but we don't know if there's a primary defect at NMJ and SMA. There may be. So from these mouse studies, we basically add this onto our clinical study because we want to see what patients look like. And that's really like, that's my MO. It's like, I have 1 foot in the lab, maybe it's a little bit of a heavier foot, and then 1 foot in the clinic. I really like the back and forth. I'm working with people that are doing primarily clinical things, and it's just, to me, I love interacting with scientists and clinicians all together. So we added this pre and post. These are the 2 papers that we just published in this cohort, and very, very strikingly, I don't know how many people do repetitive nerve stimulation here. I do about 2 patients a week, and I would say over the last 100 patients that I've done for myasthenia gravis, repetitive nerve stimulations may be positive at 10% of those. Single fiber is very sensitive. You know, if you think it's myasthenia, it's going to be positive, but rep stim is really, really crappy. It's terrible. But you can see here on average, these cohorts before and after. Now, so caveats. So this happened during COVID, and it was also started after this study had already initiated. So some of these patients had already gotten nusinersen, so they couldn't have a pre-post. So this is just cross-sectional of all the data before and all the data after. Then we have longitudinal in a subpopulation of patients. And you can see that really, I don't know, you can maybe imagine that there could be a little bit of effect in the longitudinal cohort, but it's not that overt. They still have a lot of decrement, and some of the patients have quite a bit. CMAP consistently shows improvement with treatment, and that's well established. We showed that, other people have shown that. So
there could be other interventions at the neuromuscular junction.
Exactly. So when we look at age, disease duration, ambulatory ability, no correlation, which I find interesting. That makes me think that this is not related to SMN. It makes me think that it's related to the intrinsic capacity for the individual to compensate. So there may be some people that genetically aren't able to maintain that collateral sprouting and NMJ transmission. So I don't think this is directly. So this could be relevant to other motor neuron
So
diseases.
I have friends at KU that have been trying to tell me that ALS has a neuromuscular junction component. I've been telling them they're crazy for years, but
I
they're
think
probably
so.
not.
Yeah,
And
I think
I've
so.
known that. And,
I think the problem is it's too progressive.
yeah, and Dr. Siegel will talk to you about that later.
Right, yeah.
Yeah, that's good.
And we looked at basically the correlation between this decrement and physical function. You can see some nice associations between measures of muscle strength. So this is all maximal voluntary isometric contraction. You know, I thought I had one more picture there. Well, I don't know where it went, but I had a nice picture. So the 6-minute walk test correlated really, really nicely, and fatigue on the test. So this all kind of came out of a collaboration with a small biotech company from Denmark. So Thomas Pedersen, he and I are good friends. He's a really cool muscle biologist. Now he's the CEO of this company. They have this compound that's orally available. It's being tested in myasthenia gravis now to inhibit the CLC1 channel. So that's the channel that's responsible for the majority of the threshold of muscle fiber. During repetitive firing, the chloride channel normally kind of antagonizes action potential, but during fatigue, the CLC1 channel actually gets inhibited. And so what he's doing with this compound is leveraging a normal physiological phenomenon during fatigue to improve the safety factor. And so we've been testing this in mice and other studies, and I'm not able to show the data, but that's a novel treatment. But you also have, 3,4-DAP that is out there for Lambert-Eaton, FDA approved for that. You have Mestinon. Actually just last week, somebody presented positive data on Mestinon and SMA, but it was one dose. And I actually think Mestinon's a poison to the NMJ. I think it might have effect one dose, but when you flood the junction with acetylcholine, it's been shown in genetic models that the junction gets spread out. And so I think it could have a transient effect, but I think long-term it might have a negative effect, which will balance out. I think the same thing happens with myasthenia, honestly. I think everybody takes
Or it
it.
doesn't work after about 6 months a year.
And then when they don't take it, it makes them, so it's
Right.
like this, it's just adding to the problem. Transiently, I think it works
Right.
very well. Salbutamol is something that we've actually tested in aging. Some SMA patients actually take it. And it has studied a little bit. It is an anabolic agent, but it actually has just been shown to improve NMJ transmission. So I think that is pretty much it. I don't think I need to summarize everything that I talked about too much. I will cut it a little short, but I think there's a lot of ongoing gaps, which I think are exciting. I think it's exciting that there's been so much progress in this field. This honestly is kind of like a side hobby. I don't do a lot of SMA, it's not my main thing, but you can see I'm still pretty involved with it. So I thought it might be a good topic. And kind of, I use it as a template with other diseases. And so we can apply this. Now there are some caveats. This disease is set up for success better than some diseases, but we won't go into that. So lots of people to thank. I have a really amazing lab team, clinical team. I collaborate with different groups. Most of the NMJ stuff has really been primarily in the lab, primarily with me, but then Dr. Sheik and I have collaborated in the clinic. And so I appreciate everybody's attention, and hopefully that was not too far off topic, but just gives you a sense of some of the things I've done. Okay, so the first question is whether gene therapy allows the type of modulation. Okay, now I see the full question. So the question is that genes don't return on and off, and that is actually the point of that paper that's getting everybody excited. When you deliver a gene therapy, that is, essentially kind of permanent, and you can't modulate it. It's not under the control of that cell, and so that's where you have to really find that exact perfect level. You don't want a promoter that's too strong. You don't want too big of a dose. So the way the gene therapy works is you get a number of copies per cell that are transduced, and we can measure this with different assays in the lab. And so part of the modulation is determined on how many copies are transduced in each cell, and the other is the promoter. Once it's in there, at this point, you can't modulate it. So I think that is, honestly, that's part of my hesitation with gene therapy for some things, because it's like, what if it doesn't work, or what if there's a problem, it's hard to turn off. So there are safer things, but the gene therapy is nice because it's a one-time thing. So it's almost like it's good and bad. So there's a question about SMN protein outside of SMA. Interestingly, there are a few studies with mixed findings. I think the most definitive study showed that there doesn't seem to be any association with ALS. So some people have looked at SMN 1 and 2 copy numbers and see if it changes the risk for ALS, and that doesn't seem to be the case. One idea that I have that I really would like to do is take a population that has been well characterized of like older adults and look at the prevalence of sarcopenia and see if SMN dosage 1 and 2 copies, because actually 1 can be duplicated too. Some people have more copies of SMN1. See if that predicts risk for sarcopenia. So that is something that I do hope to do. I kind of would like to do the mouse studies first to give rationale for that study, but I think it would be a very easy thing for us to test in the clinic. I think that's all the questions. Your next question.
In the room. As far as so it's a one-time dose for the AAV, and the goal is that it would effectively stay in the cell for however long. Thoughts on the idea of maybe doing an additive dose afterwards, and if you did, you would have to time it out to the appropriate amount to make sure that the immune response to that vector would, or you change the vector itself. I don't know how you would go about doing it.
Yeah, you're exactly right with that. So the question is, repeated dosing if the effect wears off with like gene therapy. And right now, like it's just not possible. So that's like exclusion. You got to screen for serologies to the vector. But would there be ways to do targeted modulation of the immune system or change the capsid or something like that? I think that's possible, but then it changes the whole approvals. So it makes it really complicated. But that is actually a really important question that is just not answered.
And then looking at biomarkers as far as the overall analysis of gene expression, we're talking about the different additive effects of having a deficient protein like SMN1 and 2, as far as like transcriptome analysis and looking at the varying expressions of everything after you give the treatment. Is that something that you would want to focus on?
Yeah, so it's a big question because during development estimate changes, and we don't know what level it needs to be when. And the biggest challenge is we think it's important in the motor neuron, and the level of blood versus like the spinal cord dramatically different. So we don't have great ways to measure target engagement in the tissue that needs to be engaged. And so that's a challenge because like, so say someone gets gene therapy and 20 years from now, the levels in motor neurons are low and they're just slowly losing motor neurons and we can't tell because they're collaterally innervating or they just think it's aging. It's like, we won't know. And so I think that's an important gap. And you know, one of the roles of SMN is probably in splicing of other genes. We don't know what low levels do to cause motor neuron loss, but that's one area where people are exploring. Like, are there different targets that are being misspliced that are causing motor neuron degeneration, or is it some other mechanism that's causing SMA?
I have a more general question. So there is a selective vulnerability for the neurons.
Yep.
First, is this random or do you have any molecular explanation for? And how the treatment will focus those in need?
Yeah, if you examine a patient, you can see different muscles are dramatically affected, like the quads are decimated, the triceps are terrible. In mice, they looked at the vulnerable populations. But we don't know what causes the motor neurons to die, so we don't know why this population versus this population is resilient or not. I think that's a cool strategy that people are trying to use. The problem is how well does the mouse replicate the human, and you can't get motor neurons that easily for humans, you know. So I think that's the challenge. And is it even in the motor neuron? Like, I think low levels in the motor neuron are what probably are driving the phenotype. But could it be in other cells? Could it be at the neuromuscular junction? Could it be somewhere else? Some people think it's an axon. So there's just so many unknowns despite having 3 therapies in the clinic, which is cool. I think, you know, and people like, you know, there for a while, grants for SMA were kind of hard to get. People like, it's done. We did it. You know, and it's not really, you know, I mean, the gene therapy is the most expensive drug in the world. So it's like, we got to make it cheaper and better and more accessible. So we're not done at all.
Well, thank you, but before we wrap this up, I want to go ahead, I always have to give a presentation, and based upon the presence of our former chair here, it was big on t-shirts, as we all know. But this is also something that's pretty easy to pick, and I told you we have something in black, I said black and gold last night.
I said I didn't have a uniform, so thank you. Great questions. It's super fun to be here.
Thank you.
Thank you.
All right, we're gonna transition now a little bit and just kind of move over to Dr. Arnold's gonna share a little bit about a personal and professional history with us. And so kind of a different way to wrap up the rest of our time that we have available. We'll have a hard wrap at a little bit before 9. And
So I didn't really plan anything for this part. We were just talking about last night. And what I thought might be of interest is just to tell you like kind of where I came from, why I'm doing what I'm doing, how I got here. 5 years ago, 10 years ago, there's no way I would have thought I would be able to do the things I'm doing now. Super lucky. It's like almost I wake up, I'm like, this is insane. I'm actually doing it. I'll just go in my lab and see my lab. It's like, this is awesome. So I grew up in the hills of Kentucky. Nobody in my family was a doctor. I thought, I want to do medicine because I'm going to have a job that I never lose my job. Because my dad worked coal mine supplies and things like that. And so I was kind of on the med school conveyor belt, as you all know. You get on the med school conveyor belt, you do pre-medicine, and you go to med school and go to your classes. And I actually went to medical school to do physical medicine. I'll get to that. So I'm a big runner, swimmer, triathlete, that kind of person. Like I didn't point it out, but the last slide is Dover in England. I want to swim the English Channel. That's my big goal. But I grew up in Kentucky, and I like sports. I thought physical therapy would be so cool, build athletes. And I had a family friend who said, "Oh, you should go to medical school and do physical medicine." He didn't know what he was talking about at all. He didn't know what physical medicine was at all. I was like, sure. And so I just was like, that sounds good. And after I read about it, learned about it, and it was like, that sounds like a really unique, cool specialty that I could do so many different things with. So I went through medical school. I thought about a few different things, but circle back to physical medicine. I did med school at University of Louisville, stayed there for residency. I was born in Alabama, but Kentucky boy all my life. I went to college like 20 miles down the road. So I was just like, it was just more convenience for me. I didn't like think outside the box, but went to med school, got into residency. I went to University of Louisville for residency. Wasn't super academic, but I learned a lot of awesome clinical experience. And the way that University of Louisville worked then is we actually rotated with a lot of specialties outside. So we worked with orthopedics a lot. It's a big hand surgery, world-renowned hand surgery team down there that I've rotated with. And kind of found that, I'm not sure I like sports medicine. And I think had I done it now, I might have been more excited about it because it's more innovative with regenerative therapies and with ultrasound and things like that. Back then I was just like, I'm just going to be dealing with whiny athletes. I'm not seeing the cool cases. And I fell in love with neurological diseases. I actually was going to do spinal cord injury first. I was like, I wanted to do that, but I wanted to do it in Louisville, but they didn't have a fellowship. But then neuromuscular medicine became a subspecialty of neurology and physical medicine, like literally the year I was graduating. And I was like, that's pretty cool. And I rotated through MDA in Louisville. They don't have a big neuromuscular program there, but I rotated through there. They had the director given us a book who actually was written by Jerry Mandel, who's where I am now. And I read that book and I was like, this is amazing. This is what I want to do. So I basically didn't know anybody in the field. I just emailed across everywhere and just said, apply for your fellowship. A lot of places, being a physiatrist, I didn't get an interview. The places that I really wanted to go, but I think it worked out. So where I ended up was OSU. My director of neuromuscular there in neurology, his brother was a physiatrist, won the Elkins Award. And so he knew kind of our skill set and how I might, be synergistic with our group. So I did the fellowship there. I kind of got more and more academically inclined, got curious. I was thinking pure clinical, but I wanted to see the complicated cases, the rare myopathies and those kind of things. And so fortunately, they created a position at OSU for me. And so I stayed on there, full-time clinic. So I was doing clinic, I had a couple clinic days. I, at that point, was running the inpatient neuromuscular consult service, so neurology would basically consult me when they had a neuromuscular patient that they were not sure about. And then did EMGs, of course. And I've always loved the sophisticated EMG techniques. And so I've always, I think I like the physiology of it. And maybe that's kind of my scientist that was like hiding underneath everything. Kind of that's the reason I like EMG. But so basically did that, and then like started interacting with people that were doing research and thinking like forward thinking, like what do I want to do and how can I get there? And I saw people doing, like one of my friends is a physician scientist. I was like, that looks pretty cool, and that would be neat. And I would love to be able to ask questions that I'm interested in. Didn't really know what I was trying to do at that point, and basically went to my chair and said, you know, can I apply for this career development award? And he said yes. And I talked to the guy that created that mouse model that I mentioned. I said, can I like work in your lab? So I basically worked nights and weekends and got preload data, then applied for the RMSTP grant, which is a career development award that gives you money to cover your time because you can't do this all the night and weekends. And then kind of transition from there. So I don't know if that's helpful or not, but I think like what I would recommend, like, don't ever think it's too late, because I hadn't published a paper until I'd been faculty for 2 years. I hadn't done research at all, and I just kind of fell in love with it. There are ways, and I think a lot of times as a physician, when we're talking to scientists, we're intimidated, but our skill set and our knowledge is something that they don't have. So it's our leg up. You have to learn the science, but you can learn the science. Techniques change all the time. And research doesn't have to be in the lab. It can be, a lot of my initial papers were chart reviews, some of my most cited papers, and some of the ones I'm most proud of. And so I think looking out, if you're a trainee looking at 5-10 years, like where do you want to be, how can you get there, and keep an open mind. I think that was the thing that fortunately helped me. And I had a lot of good people around me that I sometimes I look back and I'm like, how did I do this, and why did they let me do this? But I don't know, any questions or anybody that's interested in research, or is that like provoke any thoughts, or maybe not?
It's inspiring, that's for sure.
I can tell you kind of what I do on a day-to-day basis. Maybe that would be interesting. So I'm about 20% clinical. So my clinical time is all neuromuscular, mostly EMG. I do some outpatient consults. I don't follow a lot of patients longitudinally. Most of my time dystrophy patients. Trying to build a multidisciplinary clinic for that patient group, because I think the therapies that we have in SMA, I think there are going to be some new discoveries that are going to mirror those different technologies, but I think we're going to need the comprehensive approach like we have in SMA. We don't quite, my time dystrophy is a lot more complicated because all the other systems are involved. So clinic about a day a week. I covered inpatient consults and EMGs. I am currently 100% within neurology. So at one point I was the medical director of EMG lab at OSU, which serves about 4,000 or 5,000 EMG patients a year. So massive. That was one of my biggest accomplishments actually as a physiatrist, being like the director of the neurology lab. I was like, right. So I don't know how it is here, but it is what it is. But neurology and physical medicine doesn't always interact super strong. It's not as collaborative and synergistic as I think it could be, and that's been my goal all along. And so I think, you know, it's kind of the parallel would be interacting with scientists. Like we all have our different skill sets and different perspectives, and so building that team. That's I think one of the things I like the most about the physical medicine field is you interact with so many different people. It's like that's fun, you know. We're all a team, interact together. And in my lab like that, you know, we have different people at different levels.
We really don't have a typical week, so.
Oh yeah, I forgot. It finished my week. That's my week. Yeah, that's all. So I don't have a typical week, but so I do clinical studies. So currently I have 4 clinical trials or clinical investigative studies. So I do a lot of muscle biopsies for biomarkers and myotonic dystrophy, and we also study tendinopathies. And so we've developed a way to get muscle that's less invasive. So it's very simple. We just use a core needle. And it's kind of neat. I started doing this like 10 years ago out of necessity because we didn't have a needle device that people typically use. I don't know if you've seen a Bergstrom needle, very barbaric. I've used those too, but it's huge and you can get a massive piece of tissue. But I've been working with a group from Rochester on myotonic dystrophy because that is a CTG repeat disease. that causes basically RNA foci in the cell to be kind of like sequester RNA binding proteins, and then you get altered splicing. So a good biomarker is measuring muscle splicing changes, and so we do that with this biopsy approach. So I basically bought a steak and got this needle and just practiced to see how much I could get. And then I had a surgeon train me how to do it and I was always the medical student that hid in the corner, didn't want to do any procedures. And now I'm like, why am I doing all these procedures? But, and so I did that out of necessity because my boss told me I had to, because there was nobody else that could do it. And then I've done it. And now it's really interesting. I have, within the last 2 months, have been contacted by multiple companies that kind of design their outcomes for these genetic interventions, which is like really neat. I mean, it's a side gig, but so I think diversifying your day-to-day is fun. So clinical studies, I would say that's maybe 10-20% of my time. And then the rest is lab. And so lab, I do a lot of hands-on stuff. I have undergrad students and medical students that rotate through the lab, and then a couple postdoc fellows. So kind of a medium-sized lab. Running a lab's pretty weird because it's like running a small business, and so you're responsible for the money and people paying people salaries, coming up with ideas, and then just managing people. And so it's been very interesting, like experience to learn the leadership by running a lab, and it's very, very rewarding. But it's also, I tell people that I have a lot of students that come like, this is what I want to do. I was like, you got to make sure that you really want to do it because it's a little bit crazy. The funding, the pay for people and research is not always that easy to come by, but it's totally worth it if you love it, if that makes sense.
Yeah, I don't know if you've worked with Casey Childers much. One of our former residents and faculty member here for a while.
Yeah.
And
Went
one of
on to
our
bigger
residents did research with it.
Okay.
Yeah, asking all the in-depth
Good
questions.
question. Yeah, he's got some gene therapy background. Yeah, so I've known him for a number of years. We gave an AAPMR course together, and I'm well familiar with his dog work.
Yeah.
Really cool stuff. Yeah.
Similar to.
Started that year.
Yeah, which is really, really neat. Yeah, it's very physiologic background, kind of like nature. Very similar.
Yeah.
PhD, early 2000s here.
Yeah, so I don't have a PhD. So sometimes people, when they put me on a schedule, they'll give me one honorary. It's like it's like, no, I'm new to this. So I was like, yeah, so I do definitely like deal with imposter syndrome. It's like all these people with all these credentials and with the Kentucky boy and all that stuff. You just gotta go for it.
You went to the dark side though. He works for a big old company now.
Yeah, I do collaborate with companies a lot, and actually it is, if there's a space where you can have that back and forth, I like the academic side better, but I like how dynamic it is, and they do have resources, and it's very exciting. So, it's a little bit tricky when you're in academics, you gotta do all the things, but I really get a lot of joy out of doing that, and I feel like I contribute through that avenue too. I don't know if I would ever want to go all the way that way, but I do like interacting and collaborating. It's very cool.
Dr. Mendel's still there, I take it?
He is.
Okay.
Yeah.
Yeah, whenever I did my residency, he was a young neurologist. We had a joint clinic with the muscular dystrophy clinic with PNR and neurology together and stuff. He had already done a lot of work in muscular dystrophy at that point.
Yeah, so he was a PI on the SMA study.
Okay.
I should have mentioned that.
I'm gonna ask about ultrasound.
Yeah.
So I know you mentioned last night, you don't do it, but we teach ultrasound, a big part of our curriculum, and I was wondering what role it plays in some of this research.
Yeah.
Diversity of muscle, cross-sectional area, stuff like that.
I think.
Does it help or?
I think what's cool about these therapies is as you can change a disease, you can measure a disease and you can see the change. And I think we're very good at quantifying things and measuring and understanding function. And so leveraging all sorts of technologies in novel ways and then using the genetic interventions to see, hey, this is actually sensitive to that change. And then can this be something that we can apply in a objective manner in the clinic to quantify things. So like from a research standpoint, I think definitely, but then clinically, very much so too, like peripheral nerve injury for sure, quantifying disease progression or severity. Possibly, I think as technology gets better, as we learn to do it more in a more repeatable way, it's gonna get better and better. So I love biomarkers and measuring things. I think that's one gap for our field is measuring things and understanding what impact we're having with our interventions. Because you know, we want it to work, and so if you don't have something that's objective, sometimes you don't really know how much it's working unless it's very objective. So I like biomarkers.
Yeah, something I've been learning about and trying is measuring the echogenicity of the muscle and actually the pixels, and they can give you the grayscaling.
Yeah.
And that's been correlated with strength and things like that. So
Right.
I think that's really exciting, and
And-
I like that area.
And what if we did it across, what if we didn't stick to one disease? We just did irrespective of disease,
Yeah.
and see how it works for all our rehab patients,
Yeah.
you know? And our interventions and our exercise. And is there a way to quantify like contraction, not just static? Dynamically, can we see the shortening and things like that? You know, there's a lot of, like, think outside the box. You know, there's a lot of like low hanging fruit that, go for it. You know, I think that research is fun, even if you don't want to focus on research. It adds to your clinical expertise. I think like we're taught to just kind of learn and then regurgitate, but I think being critical and pushing the envelope is very rewarding. Like it really is fun, and our patients love to be involved. They're doing research. I can't believe how many times they come in and they're just so excited. It's like, the study doesn't actually have an intervention. I'm just measuring stuff, you know, I'm taking muscle, and they're just so excited and so willing. It's just like so much of a team aspect. Like, I feel like clinical research, I get like a lot of the good stuff in clinic, but in the research setting, it's pretty fun. I do a lot of clinical studies, but more.
Payback that the patient can get.
Yeah,
Right,
Right,
And
yeah.
neuromuscular problems across the board. I find them also to be just very giving.
oh, for
They
sure.
wanna, you know, and you explain to them, you know, this may help you in the shorter or in the longer term, but it may help a greater number of people. And they just seem to be very giving in that arena.
Yeah.
In the neuromuscular arena, and sort of the spasticity work that I've done. They're very good about that. Questions, guys? This guy's got a busy day. We've got a
Yeah,
couple more
yeah.
minutes with him.
I have a break between 6 and 7. Literally, 6 and 7.
Yeah, that's it.
Yeah, I normally get up really early, and today I'm like, no, I'm gonna sleep.
Is that when you train?
It's always been my like time to get it in, so
Self-done.
they can't take it away from me. Get up early enough. I know when I was doing neurosurgery and residency, it was like 3 o'clock. It was like, oh. But I had to get it in, you know, I want my run or whatever.
You either do or you don't. End of days get pretty
It's not gonna
variable, so
happen.
beginning of days.
And I have
you set
kids,
your-
so I like to be done when
And
they're not awake at 4.
they're not. Go figure.
My son sometimes will get up and it's got one of those Oculus. Gets early to play that thing. So. A little bit of dad in him, I think.
That's awesome.
Thanks again.
Thank you.